Good morning, good afternoon, and good evening. My name is Leilani Marti Santos, and I am a field application scientist at Akoya. Today, I will be presenting a beginner's guide to Codex as an introduction to the Codex workflow. In this webinar, we will be covering spatial biology and the need for single-cell resolution, the Codex chemistry and workflow, a Codex case study including data analysis examples, and at the end, we'll have time for questions and discussion. First up is a look at why we need true single-cell resolution, particularly when we're discussing spatial biology. The saying goes that no man is an island, and as biologists, we know that no cell is an island. This is a cartoon of a tumor microenvironment that you may have seen in the first talk of this webinar series. My colleague, Clemens Durschmidt, rightly said that no cell exists in isolation, and while this cartoon is an example of a tumor microenvironment, this could easily be applied to any tissue that is being studied. I'd like to break this cartoon down further and start by pointing out that even when we're focusing on the example of a tumor microenvironment, around 50% of the cells in that microenvironment are non-tumor cells. You can see that the malignant cells interact with the extracellular matrix and with cancer-associated fibroblasts. Their interactions with both vascular and lymphatic endothelial cells. A whole host of different immune cells, represented here by helper T cells, killer T cells, B cells, NK cells, dendritic cells, and macrophages, and other cells that reside in many tissue compartments, such as red blood cells, adipocytes, and mesenchymal stem cells. While this image is an extremely simplified view of the microenvironment, we can already observe 14 distinct cellular phenotypes, and don't know how many other cell types, including rare cells that have yet to be identified, may be missing in this diagram. Every cell depends on its interactions with the many other cells and cell types within a tissue. This serves to underscore why trying to answer biological questions with only one or two markers in isolation and/or without retaining the spatial context in which a cell exists ultimately offers limited information and therefore limited predictive value on the path to true precision medicine. When we look at patient samples, we often see the use of immunohistochemistry on formalin-fixed paraffin-embedded tissues. This traditional method allows for nice spatial resolution to the point of being able to identify single cells expressing a particular marker, but it is limited by low dimensionality, meaning that it requires the use of single markers at a time. When you have precious tissue samples, this can be extremely limiting. On the other hand, single-cell analyses, such as single-cell RNA sequencing and CyTOF, have become very popular and allow for high-dimensional analyses of hundreds of transcripts and/or proteins. Different cell types are color-coded on this t-SNE plot, but you lose the full picture of what's going on within the context of the tissue itself. With the Codex system, however, you retain the spatial context along with the capability to generate single-cell information with a high number of markers, 40 markers or more. Although this image only features 6 markers, It and the Tisney plot featured here are from a 36-plex breast cancer Codex experiment done by our research and development team at Akoya, and I'll be using these data as an example throughout my talk. To show you why true single-cell spatial biology is so powerful and important, I would like to demonstrate this on an average size piece of tissue. In this case, we're looking at a section from a stage 2 breast cancer specimen. Each cell can be readily identified by its nucleus via DAPI staining. However, if we use a region of interest grid-based approach, we lose almost half of the data. The spatial information is partially lost and features are disconnected. On top of that, region of interest grid-based methods average the biomarker expression for each region of interest, thereby losing even more resolution. We are unable to accurately conclude what is happening in the tissue spatially, if multiple different cell types interact within each of the regions of interest, and even less so how they interact across regions of interest. This is a comparison of two different images, looking at the expression of CD4 in a tissue. When we look at an example of single-cell multiplex spatial biology, shown on the left, we notice that there are some very highly expressing cells across the tissue, but they are few and far apart. When we use a region of interest grid-based approach, shown on the right, we don't even notice these cells are there. At best, this is a spatial approximation. The power of single-cell spatial biology lies in the ability to address each cell uniquely. Before I go further into the data generated by our R&D team, I'd like to highlight the Codex chemistry and the workflow behind a Codex experiment. The name Codex comes from code detection by indexing, in which we use antibodies conjugated to proprietary Akoya barcodes, comprised of a unique oligonucleotide sequence. The Codex assay targets specific barcodes with a fluorophore-labeled reporter for highly specific detection. The idea behind the Codex platform is that Akoya offers the beginning-to-end solution for your highly multiplex data acquisition. We offer inventoried barcoded antibodies, in addition to selling Codex reporters, and custom barcodes that allow you to conjugate your own markers of interest that you already use in your lab and include them in your multiplex panels. We have an automated fluidics platform that integrates with different microscopes, and we have a software suite that controls the imaging and the fluidics and also allows for the subsequent analysis of the images generated. The staining of your tissue relies on the use of Codex antibodies that have been conjugated to unique oligonucleotide barcodes. Because we sell these unique oligonucleotide barcodes a la carte, you're able to conjugate your own markers of interest that you may have been using in your laboratory, in addition to using the inventoried Akoya antibodies that we have in-house to create a highly multiplex panel. Then, during imaging, reporter oligos that are conjugated to fluorophores bind to their complementary barcode. So how does a Codex run work? The first step, which is the only manual part of the protocol, is the tissue preparation, as you would expect for either immunohistochemistry or immunofluorescence. If you're using formalin-fixed paraffin-embedded tissues, you would follow a deparaffinization protocol, followed by antigen retrieval. And if you're using fresh frozen tissues, they would be allowed to thaw to room temperature prior to fixation. Then you begin the staining protocol. The main difference with the Codex is in staining your tissue, which is done by applying all of your barcoded antibodies at once, whether you're using 4 or 40 markers. We then turn to our Codex Fluidics instrument, which is a device that automatically handles liquid dispensing and integrates with inverted fluorescence microscopes via a common control computer and a Codex Fluidics stage insert. Once the tissue is loaded into the Codex stage insert on your microscope, we start the revealing process, in which the Codex applies 3 reporters at a time, plus DABI, to be able to observe where 3 of the markers are on the tissue, as these reporters will only bind to the antibodies we intend them to. The microscope will excite the fluorophores conjugated to these reporter oligonucleotides and image the tissue. The Codex machine then does a series of gentle isothermic washes that removes those 3 reporters. And the Codex system repeats this process over and over until all markers have been imaged. As an example of the images generated within a Codex experiment, here I'm showing a smaller Codex panel, consisting of 15 different markers on the same tissue section of a Codex cancer sample. You can clearly see how different tissue features are highlighted in each of the images while still maintaining tissue integrity across multiple cycles. The third part of the Codex system is our software, the Codex Instrument Manager, or CIM, which controls the fluidics and the microscope as a virtual user during acquisition, the Codex Processor, which performs drift compensation, deconvolution, and other algorithms that ultimately allow for cell segmentation and the generation of single-cell data, and the Multiplex Analysis Viewer, or MAV, which allows for visualization and subsequent analysis. In addition to the Codex software suite, we also offer VisioFarm, which integrates with the MAV and is able to work with the Codex data, or as another option, you are able to use your own analysis pipelines for subsequent analysis once you've done your Codex experiment. Now I'd like to go into detail using the 36-plex breast cancer experiment done by Akoya Research and Development as an example of what the Codex is capable of. These are the markers used in the 36-plex antibody panel that I showed an example image of at the beginning of this webinar. You can see that there are multiple markers for the tumor and for epithelium. Several oncogenes were also used in this panel, as were multiple immune markers. And then in addition, there are several markers used to either highlight the breast cancer tissue itself, proliferating cells, and other types of cells within this tissue. However, even with that list, at Akoya, we believe in the power of images. You can probably appreciate the details and the variety of cell types labeled much more as I go through different overlays of the markers, ranging from 5 to 7 markers on each image in our panel on the stage 2 ductal adenocarcinoma sample. As we move through the images, you can see very different features and a different appearance of the biological system. Knowing that we used to do this 3 antibodies at a time is now almost unthinkable and serves to truly highlight the capabilities of the Codex system. As I showed earlier, Codex takes into account the nucleus of every cell using DAPI for segmentation purposes within the Codex multiplex analysis viewer, or MAV software. Each nucleus is outlined and will have a unique identifier. In this case, 63,056 cells were segmented within the tissue. So you can see that these identifiers range from number 1 all the way down to 3 to 63,056. Every cell in this assay will have different fluorescent intensities that are associated with it for each of the 36 markers in this panel. However, unlike other single-cell techniques that rely on dissociated tissues, our tissues are intact. And that allows us to retain the X and Y coordinates for every single cell. This is an example of the data that is generated by our MAV, although this data can also be transferred to other analyses pipelines such as Visiopharm. In this case, we will continue with our analysis of these data within the MAV to perform unbiased clustering to look at the single-cell biology. These are t-SNE plots, which have taken the tabular form of the data shown in the previous slide and sorted and clustered it in different regions based on the expression patterns of different cells. Each one of these plots has the same regions and shapes to it, and each graph encompasses the same 63,000 cells we saw listed earlier. What varies for each plot is the expression or intensity of a specific antibody, where red indicates strong labeling and blue is associated with weaker labeling. As an example, if we look at this region here on the left, we can appreciate the very strong staining for certain keratins, specifically keratin 17, keratin 5, and keratin 14. By contrast, we can look at this other region on the right that is strongly labeled by 3 different keratins, namely keratins 19, keratin 18, and keratin 8. We can see that for the cells on that left, there's very strong staining of these specific keratins and a couple of other markers, with weak staining for most of the other antibodies. For the region on the right, we can also see specific staining for a couple of other keratins and a few other markers. Since we know from the literature that these specific staining patterns are associated with basal cells and with epithelial luminal cells, as users, we can annotate the information within the MAV based on these expression patterns. And we can continue to annotate the different populations identified in the analysis based on the single cell information obtained through Codex. With this information, we can move into single-cell biology with single-cell resolution. While we don't have spatial information from this t-SNE plot, we can toggle back to the histological images from which we started. Here, we focused in on a region of interest from our original image, and you can see as an example the varying expression levels of the marker E2F1. It's widespread throughout the tissue, but you can see that there are cells that express low levels of this marker, where there are other cells that express really high levels of this marker. However, we would like to see the basal and luminal cells within the tissue. We can have MAV map where the basal cells, shown here as dark blue dots, and where the luminal cells, shown as cyan dots, are located. The power of Codex and MAV is that you can also move back and forth between the cluster data and the morphological data. Here you can see the expression of keratin 8 in cyan. Keratin 8 is a marker of luminal cells, so this is nice corroboration of our cluster labeling, as is a strong expression of keratin 14 in dark blue for the basal cells. When we dig deeper and combine the clustering and morphological data, you can see how the basal cells are only in the outermost layer of the epithelium as expected. However, we can see some epithelial labeling in cells that aren't considered part of the luminal population. So you can see these cells here that aren't labeled by that keratin 8 or by these cyan dots. So what are these cells? To identify that, we can draw a region of interest to select the cells within that region, and then move back to the clustered data to determine what these cells are. Although for now we're going to call this little yellow population a rare luminal cell, and we'll see what they're stained with. When we actually look at the staining pattern, we can see that these cells are strongly labeled with beta-catenin compared to other cells in the tissue, and have been assigned to a separate population based on this. This type of cell had been previously described in single-cell RNA-seq from another group, and is an extremely rare cell type. You can see here that there are 44 in total of the cell type in the tissue. This means that the Codex system allows us to very precisely and reliably recapitulate results seen by other groups using different methods. As a final example of data analysis, let's look at spatial interactions. The MAV software can measure interactions between two types of luminal cells. In this case, the L1 and L2 populations, the basal cells, and those rare luminal cells within a given distance. In this case, we're looking at between 5 and 35 microns. The main types of luminal cells expressing keratin 8 for the most part interact with each other, indicating intermixing with each other in the tissue. Interestingly, though, when we look at the rare luminal population, these rare luminal cells do not interact with the main luminal cells. They actually associate only with basal cells. This rare population is an inner epithelial layer that exists next to the basal layer in a completely different ductal structure. This discovery by our research and development group highlights the power of the Codex system for discovery using single-cell spatial resolution. In summary, for this case study, you've seen a 36-marker Codex experiment done on human formalin-fixed paraffin-embedded breast cancer tissue that has been used for single-cell analysis with reliable and unbiased cell phenotyping using the k-means clustering algorithm within the MAV, showing that we can achieve single-cell spatial resolution on an intact tissue using the Codex platform. As a takeaway, I want to highlight 3 main points. Single-cell spatial resolution is critical for understanding biological questions. Understanding the properties underlying individual cells in an environment, whether within a tumor, during homeostasis, or a developing tissue, requires also being able to visualize the other cells they interact with within the tissue. The Codex system integrates with any inverted microscope, and the workflow is simple. We have users that have paired the Codex with different types of microscopes within the laboratory, and the workflow is based on a single-step application of all your antibodies at once, followed by the automated imaging process. Codex allows for high-level multiplex analysis while retaining spatial context. Our workflow currently allows for the use of more than 40 markers in a single panel, and your morphological data is integrated with your single-cell data. And with that, I'm happy to take any questions.
Thank you so much for attending today. We will now open the floor for questions. Please utilize the Q&A tab at the bottom of your screen. And also today's slides and webinar will be available at the Akoya Academy website. Also, if your question isn't answered today or you have questions at a later date, please email us at support@akoyabio.com. So our first question is, how many targets can you analyze in parallel?
That's a great question. So as of now, we have more than 40 barcodes available. I think we have between 46 and 48 available. So we're able to look at that many targets at the moment. However, we continuously have barcodes coming from R&D that have been validated, and so that number will continue to increase.
When we're referring to repeated washings, do they cause loss of antigen in subsequent cycles?
You actually don't lose any antigen, so everything has been bound. Our process includes some fixation steps and everything that allow you to maintain everything. We have groups that are actually using our Codex system for very large tissue analysis with high number of targets that they're looking at. And so their runs go for over 3 to 5 days sometimes. And so they actually don't lose any other signal or antigen in those processes.
Okay, questions about software. Are MAV and Visiopharm open access, and can Halo software be used for data processing?
So in our case, so MAV is open access. It's actually a Fiji plugin. So if you've used ImageJ before, you can download the plugin and work from there. Visiopharm is not. Visiopharm is a paid software. It's going to be similar to Halo. And so in any case, a lot of these images, especially things that are coming from our Codex users, they access a variety of pipelines for both processing and analysis.
Great. So when referring to the tissue remaining intact, does this mean it can be used, for example, RNA isolation after a Codex cycle?
So yes, we actually have users that use the tissue for different types of in situs after the Codex process. We also have users that use the tissue for H&E or other histological stains after they've done their Codex experiment. You're also able to rerun your Codex experiment if you decide that, for example, you would like to increase exposure times or just kind of continue to optimize your images. So you are able to use your tissue in more than one analysis.
Can I use the CIM in my own microscope computer? I have an Axio scan from Zeiss. Its core microscope is an Axio observer.
So SIM installation usually will depend on a case-by-case basis. We do have some basic parameters that we include whenever we are talking to potential Codex users. And we provide them that basic information, what our basic requirements are. So we might be able to run the SIM on your own computer,
but it really will just depend on what the parameters are, and we can share that information with you after this webinar.
Can you visualize any of the stains on a standard lab microscope or only the Codex system?
So the beauty of the Codex system is that it actually allows you to connect the Codex box itself to many different microscopes. So we have users that are using Cannes microscopes, Zeiss microscopes, Leica microscopes. So you're basically able to use this on any inverted microscope right now. We also have a new Codex open platform that is opening up to allow the use of Codex with other microscopes. We also have our innovators group. So feel free to reach out to either customer care or support at Akoya to see if your microscope is compatible with the Codex system, and we'll be able to go from there.
How large is the data output of a Codex cycle?
That's going to be variable depending on how many markers you're using and how big of a tissue you're imaging. We have data that goes anywhere from a few gigabytes to up to a terabyte, depending on just how much is being imaged at once and how many markers are being used.
Is the microfluidics exchange to the microscope manually done?
So no, that's one of the key components of the Codex system. So while your staining process is the only manual portion, well, you'll be actually staining your tissue at the wet bench in the laboratory, everything after that is done using the automated fluidics platform. So once you fill in your reagents on the bottles that sit next to the Codex system, basically the Codex takes over. So that's where the SIM, so that Codex instrument manager, is going to take over. So it's going to automate the fluidics, control all the fluid dispensation and aspiration of all your reagents. And then it's also going to serve as a virtual user for the imaging portion of the acquisition.
Can this technique be applied to frozen tissue? And if so, how many markers can be used?
Yes, this technique can actually be applied to both FFPE and frozen tissues. And it's the same number of markers. So you have the ability to access more than 40 markers for the frozen tissues as well. We actually have the same barcodes available for both FFPE and fresh frozen and can go from there.
Can you analyze different regions of interest separately, or is it a whole image analysis?
So you can do both. We have users that choose different regions of their tissue that they're really interested in. We have people that are actually working on TMAs, so tissue microarrays, where they're selecting which of the cores that they're looking at. And then we have groups that are using the whole cover slip. So they're taking basically a full biopsy sample or a full section of tissue and then analyzing from there.
Are we able to stain the tissue again at a later time? For example, a month later, if you decide to look for more interest, to look at different proteins, or do we need to decide from the start?
So you would need to decide your panel. So you're able to do subsequent H&E and other types of histological stains after the Codex experiment is done, but for your actual panel of markers, so whether you're using 4 or 40 markers, those need to be decided ahead of time. So you're not able to restain once you've done the full Codex staining protocol.
Can we quantify expression of markers?
Yes, so you're actually able to look at not only how they're expressed, but you can look at relative expressions. If you look at how the output is done, you not only get your relative data in terms of your X and Y, which is lost in a lot of other types of analyses, but you're also able to see what the relative intensity is of your different markers across your different cells. So in the example I gave during the talk, we had 63,000 different cells, and you're able to get your expression for each of those cells within, for each of the 36 markers, for example, that we presented.
How is consecutive imaging done? Is there a cover slip removal after every scan?
No, that's the beauty of the Codex system. You're able to continuously image. So what is done is that you're gently floating 3 reporters at a time. You have these gentle isothermic washes that will then remove those reporters after they've been imaged, and then you continue on with the cycle. So 3 reporters at a time, they get gently washed off after they've been imaged. So you don't have to move your cover slip at all. Everything is automated, and you just let the system go to the end of your experiment, whether it's a 3-hour experiment or a 3-day experiment. It just continues on automatically.
All right, thank you for joining today's webinar. Again, please send any questions you might have down the line to support@akoyabio.com, and the webinar will be available at the Akoya Academy website.